previous class we discussed about glycolysis and pyruvic acid oxidative decarboxylation and Krebs cycle. So, in these three steps the glucose is oxidized into carbon dioxide and different steps and different compounds are formed. In different steps the NADH2 and FADH2 they are synthesized or reduced, and by substrate level phosphorylation also some ATPs are produced. Here NADH2s and FADH2s, so these are here they are having the energy produced in glycolysis and oxidative decarboxylation and Krebs cycle. So, the reduced compounds oxidized in the fourth step. The fourth step is Electron Transport System, also known as ETS, Electron Transport System or ETS. So, the first step glycolysis, it is taking place in the cytoplasm. So, in glycolysis 2 NADH2s are produced. And the second step is pyruvic acid oxidative decarboxylation. In this step also 2 NADH2s are produced. And in the third step, Krebs cycle. In Krebs cycle at 3 points 6 NADH2s and at 1 point 2 FADH2s are produced. So, all these reduced compounds are oxidized in this step. So, the fourth step is taking place in the mitochondrial inner membrane. So, here it is mitochondria, it is mitochondrial matrix. Next, here between the outer membrane and inner membrane, the space is called as intermembrane space. Intermembrane space. Next, inside the inner membrane, it is inner membrane. Inside the inner membrane, it is called as mitochondrial matrix. So, in the three regions, it is membrane, it is inner membrane. And outside the inner membrane, it is called as intermembrane space. So, this space is intermembrane space. Next, mitochondrial matrix. In the three regions, the electron transport and proton translocation is taking place. So, in the electron transport system, ETS or electron transport chain, ETC, here some enzyme complexes are involving. So, the enzyme complexes are present on the inner membrane. So, these are the enzyme complexes. First complex, second complex, third, fourth. These four complexes are involving in the electron transport. The four complexes are, first complex is NADH dehydrogenase. And the second complex is succinate dehydrogenase. Third complex is cytochrome C reductase. Cytochrome C reductase, also known as cytochrome B C1 complex. The fourth complex is cytochrome C oxidase. It is also called as cytochrome A3 complex. It is the fifth complex. Okay, in electron transport system, only 4 complexes, 4 protein complexes or enzyme complexes are involving. These enzyme complexes are present on the membrane. The first complex, it is exposed to mitochondrial matrix and intermembrane space. But here the second complex is it is attached on the membrane. Here it is exposed to only mitochondrial matrix. Now it is not exposed to intermembrane space. And third complex is having cytochromes, cytochrome B, cytochrome C1. Fourth complex is having cytochrome A, A3, and 2 copper proteins. So these 4 complexes are involving in the electron transport. Here 2 mobile electron carriers are also present on the these are present on the membrane. So one is ubiquinone and the second one is cytochrome C. Here ubiquinone is it is present within the membrane. It is movable, so it can transport the electrons from one complex to other complex. So it is moving, mobile electron carrier. Cytochrome C also, it is moving, it is attached on the membrane, it is exposed to intermembrane space only. So here it is moving along the membrane outside only, or towards the intermembrane space it is moving. And it is the 4th complex. Here 4 complexes are involving in the electron transport system and 2 mobile electron carriers. Next here, if you see the 3 steps, in the 3 steps some NADH2s and FADH2s are synthesized or reduced. So the mitochondrial NADH2 means the second step, pyruvic acid is here undergoing oxidation and decarboxylation. So in the second step 2 NADH2s are produced. So these NADH2s are produced in the mitochondrial matrix only, and in the Krebs cycle is also taking place in the mitochondrial matrix. So the second and third steps some NADH2s are produced. So total in second and third steps 8 NADH2s are produced. So this mitochondrial NADH2, it is mitochondrial NADH2. Mitochondrial NADH2 present in the mitochondrial matrix. So the NADH2, when it is oxidized, it is giving electrons to this complex. Here 2 electrons are given to first complex. First complex is NADH dehydrogenase, or the first complex in the presence of this enzyme, the NADH dehydrogenase. In the presence of this enzyme complex, here NADH is oxidized, reduced in the here 3 steps, but here it is oxidized. The mitochondrial NADH2, when it is oxidized, electrons are released. These electrons are given to the FMN, flavomononucleotide. So first complex, it is taking electrons from NADH2. So it is oxidized, it is reduced, and this first complex is giving electrons to ubiquinone. The electrons are moving from NADH2 to first complex to ubiquinone. So when the electrons are moving from this NADH2 to first complex, the protons are also moving, hydrogens are also moving from mitochondrial matrix to intermembrane space or outer space. Here 2 protons are also moving from mitochondrial matrix to intermembrane space. Next here, the electrons are here received by ubiquinone. The ubiquinone is now here it can move from one place to other place. So it is taking electrons from this complex, and the electrons are given to the third complex, not to second complex. Here it is taking electrons from first complex, the electrons are given to third complex. Third complex is cytochrome B, cytochrome C1 complex. Next here the third complex is cytochrome C reductase. Means the enzyme is involving in the cytochrome C reduction. So it is giving electrons to cytochrome C. It is taking electrons from ubiquinone, and it is giving electrons to cytochrome C. Means here it is oxidized, it is reduced. So after receiving the electrons, the cytochrome C is reduced. Means it is involving in the reduction of cytochrome C. That's why it is named as cytochrome C reductase. So So it is reducing the cytochrome C. Next, cytochrome C, it is moving, it can move on the membrane, inner membrane towards this intermembrane space. Cytochrome C is giving electrons to the fourth complex. Fourth complex, cytochrome A3 complex. So when the electrons are moving, here when two electrons are moving from NADH2 to FMN or first complex, here two protons are moving from mitochondrial matrix to intermembrane space. So protons are translocated, two protons are translocated. Next, here also two electrons are moving from here first complex to third complex. Then also here it is ubiquinone is taking two protons from mitochondrial matrix, and these protons are given to or released or translocated into the intermembrane space. So ubiquinone is taking electrons from first complex. These electrons are given to third complex. It is taking protons from mitochondrial matrix, and they are translocated to the intermembrane space. Here, just the ubiquinone function is taking electrons and donating. It is donating the electrons to third complex. Next, protons from mitochondrial matrix to intermembrane space. They are translocated. Next, here also two protons in the electrons are moving from third complex to fourth complex. Here also two protons are moving from mitochondrial matrix to intermembrane space. So the electrons are here. Electrons released from NADH moving from first complex to ubiquinone, third complex, cytochrome C, fourth complex. At last, the electrons are received by half molecule oxygen. So it is taking electrons from cytochrome A3. So two electrons and two protons from mitochondrial matrix. Here water is formed, one water molecule. When one NADH2 is oxidized, oxidation, when it is oxidized into NAD plus, here electrons are released. The electrons are given to first complex, next ubiquinone, third complex, cytochrome C, fourth complex. At last, the electrons are reaching the oxygen. So in the presence of oxygen only, here the total aerobic respiration is taking place. In the previous steps, first, second, third steps, the oxygen is not involving. Oxygen is not involving. Here oxygen is present and it is involving in the fourth step only. So in the three steps, oxygen is not directly involving, but the oxygen presence is compulsory for aerobic respiration. So in the presence of oxygen only, the aerobic respiration is taking place. But here, oxygen, it takes electrons from the fourth complex and protons from mitochondrial matrix. Here water is formed. So when one NADH2 is oxidized, one water molecule is formed. So electron movement is like this. It is a electron transport system. So in the electron transport system, here these complexes are involving. Next, what about FADH2? FADH2 is also synthesized in the mitochondrial matrix during the Krebs cycle. So the FADH2, when it is oxidized, here the electrons are given to second complex, and the electrons are here reaching the ubiquinone. So let us see here FADH2. When FADH2 is oxidized, here FADH2 is synthesized in the Krebs cycle. So So FADH2 is oxidized to release electrons. Here 2 electrons are released or transported to second complex. The second complex is succinate dehydrogenase enzyme. So in the presence of this enzyme, here electrons are, the FADH2 is oxidized and electrons are released. So these electrons are received by the second complex, and here electrons are moving to ubiquinone. So when FADH2 is oxidized, the first complex is not involving. First complex is not involving, only here second complex ubiquinone, third complex cytochrome C, same process. But first complex is not involving. When NADH2 is oxidized, second complex is not involving. When FADH2 is oxidized, only here second complex ubiquinone, third cytochrome C2, here fourth complex, next oxygen. So electrons are moving in this manner, same process. But here when it is oxidized, only 2 protons, here 2 protons are moving. When NADH2 is oxidized, total 6 protons are translocating from mitochondrial matrix to intermembrane space. But when FADH2 is oxidized, only here 2 protons here and 2 protons here, total 4 protons are translocated. So here the material which is oxidized in the electron transport system or in the oxidative phosphorylation, so the material which is oxidized, so the based on this material NADH2 or FADH2, so based on this material or based on this compound only, the number of protons are translocating. Here NADH2 is oxidized, 6 protons, FADH2 is oxidized, total 4 protons are translocating here. So total here the 4 complexes are involving in the 4 enzyme complexes are involving in the electron transport system. Next, what about glycolysis NADH2? So we discussed only a mitochondrial NADH2 and FADH2. But glycolysis NADH2, when glycolysis NADH2 is oxidized, here the glycolysis NADH2 means that is taking place in the cytoplasm. So glycolysis NADH2. The glycolysis NADH2 from cytoplasm to mitochondria it enters. Here NADH2 it is produced in the glycolysis. The site of glycolysis is cytoplasm. So the NADH2 is present in the cytoplasm. But here during the electron transport system or oxidative phosphorylation, it enters into the mitochondrial matrix. So the glycolysis NADH2 here it directly gives electrons to ubiquinone. Glycolysis NADH2 or cytoplasmic NADH2, it gives electrons directly to ubiquinone. So here in this electron transport system, only ubiquinone is involved, ubiquinone and third complex cytochrome C, fourth complex, here oxygen. So the electron movement is like this up to oxygen. So from ubiquinone it is common for all, whether it is glycolysis NADH2 or mitochondrial NADH2 or FADH2, here it is common for all, from ubiquinone, third complex, cytochrome C, fourth complex up to oxygen. But here ubiquinone is receiving electrons from first complex, sometimes here second complex, or from here glycolysis NADH2. So depending on this NADH2 or FADH2 or glycolysis NADH2, here the number of protons translocating it is depending. Here when glycolysis NADH2 is oxidized, total 4 protons are translocating, and FADH2 is oxidized, here also 2 protons, 4 protons are translocating. When NADH2 is oxidized, here 2 protons, total 6 protons are translocating into the intermembrane space. So it is about electron transport system. Next, what about oxidative phosphorylation? So for oxidative phosphorylation means here the synthesis of ATP here during the oxidoreduction steps. Here oxidative phosphorylation. So for oxidative phosphorylation, here the proton concentration. So based on the proton concentration, the ATP synthesis is depending. So let us discuss about oxidative phosphorylation.